HBCM Lectures on Musicians' Hand Dystonia. Author Joris Lijnse. Director Hand-Biomech-Ctr for Musicians. This lecture was published at Zenodo. Hello, my name is Joris Lijnse. I have established and direct the Hand-Biomech-Ctr for Musicians in Belgium. The HBCM intends to produce a series of lectures on musicians' task-specific hand dystonia. This is the first lecture. The lecture presents a theoretical model of how peripheral factors may trigger the condition. The next lectures will present case studies demonstrating and analyzing peripheral factors in individual musicians with hand dystonia. Lecture 1. Etiological multifactorial model of causative peripheral factors and processes in musicians' hand dystonia. The contents of the lecture are following. After viewing some musicians' hand dystonia examples, a brief review of current research and treatment is provided. Then the model is presented with supporting anatomical facts. The model itself consists of scenarios differentiated by factor configurations. The relevance of the model for treatment, including the limitations in its applicability, and some other aspects such as the unpredictability of outcomes of current treatments are discussed. Part 1. Introduction, State of Research and Treatment, Peripheral Factors, Basic Facts, Factor Analysis of Playing. Why these lectures? Decades of neurological research did not explain the causes of musicians' hand dystonia. Some musicians recovered, others managed with symptoms, but most do not fully recover. The results of any treatment remain unpredictable. In this lecture, a peripheral factor model is presented of how individual peripheral factors can be necessary cofactors in musicians' hand dystonia and determinant of the likelihood of recovery. The peripheral factor model forms the theoretical framework that will guide the analysis of peripheral factors in the case studies presented in the follow-up lectures. A brief bio may motivate my interest and experience in musicians' hand dystonia. I am myself a musician's hand dystonia survivor with multiple dystonic episodes and writing problems. At the Conservatory of Brussels, Belgium, I quit my violin studies because of left-hand dysfunctions. Similarly, I quit a subsequent study in graphic arts because of right-hand issues. I switched to studying engineering in applied mathematics, but again developed right-hand problems in handwriting. Therefore, as an engineering student, I started studying the condition, teamed up with a hand surgeon, and in a period of 10 years had multiple biomechanically adverse anatomical variations surgically removed from both hands with relief of the dysfunctions. Based on this research and results, I set up a multidisciplinary hand research lab which I conducted at departments of hand surgery, biomechanical engineering, and anatomy at various institutes and countries. I took my PhD entitled Finger Exercises with Anatomical Constraints with maximal honors at the Erasmus University of Rotterdam and held specialized research professorships in hand biomechanics and anatomy in the United States and in Belgium. Recently, I founded and currently conduct a Hand-Biomech-Ctr for Musicians in Belgium. For publications, see the website of this center. When I started studying peripheral factors in musicians' hand dystonia, I found the basic knowledge in biomechanics and functional morphology insufficiently specialized. Therefore, to lay solid scientific foundations, I conducted with collaborators and students many years of basic research in hand biomechanics, hand anatomy, reconstructive hand surgery and hand control, including development of new hand function measuring devices, software, and measuring protocols. This multidisciplinary research is as the human towers in Barcelona in this picture. Consider the explanation of musicians' hand dystonia to be the person that has to be put on top of the tower. To achieve this, hundreds of people have to provide a foundation on which the tower can be erected. This specialized fundamental research explains why all presented materials were generated in my specialized multidisciplinary hand research lab, including the dissections and their photography. However, this research lab and its productions were only possible by the collaboration and support of administrators, colleagues, students, and technicians at the various departments and institutes where the hand lab was embedded. The main institutes and support are listed here. The basis of the task-specific musicians' hand dystonia model here presented was published in Biological Cybernetics in 2015. The co-authors are Mark Hallett, neurologist, movement disorder specialist, chief of the Human Motor Control section of the National Institute for Neurological Disorders and Stroke of the National Institutes of Health, Bethesda, USA, and Jan Sonneveld, plastic and hand surgeon, with whom I collaborated and who set up a hand clinic for musicians when I was at Erasmus University Rotterdam in the Netherlands. Two examples are presented of task-specific hand dystonia in professional musicians. The first example is the right hand of a concert accordionist who now cannot even play a simple scale. The second example is a flutist's right hand, which cannot play at all anymore. The examples show what a debilitating condition musicians' hand dystonia can be, ruining performing careers. The diagnosis is symptomatic since the causes are unknown. A hand-finger coordination problem, generally painless, that occurs only in playing, with normal hand function in other tasks. Similar to writer's cramp, it is also called music cramp. While the mean onset age is 33, it can start much earlier at 24 or 25. Resistant to therapy, it frequently ends a performing career. Musicians often report trigger events preceding the onset of symptoms. This will be discussed further. Musicians' hand dystonia is classified as a focal task-specific movement disorder, occupationally induced, and also as an overuse syndrome since it occurs only after many years of intensive practice. The prevalence of musicians' hand dystonia is more or less 1% of professional musicians, strongly depending on the instrument type. There is an extremely strong correlation between the affected hand site and the instrument type. The reported gender ratio is 65 to 75% male. However, the clinically reported gender ratios may not be the actual gender ratios. Reported are the gender ratios in musicians' hand dystonia seen at clinical centers. However, these ratios may not reflect the real dystonia gender ratios. Musicians' hand dystonia gender ratios should be determined relative to the male-female ratios of the musicians' populations, which differ per instrument. For example, a 2014 survey found a gender ratio of 63 to 37% male-female players in US orchestras. Therefore, if the real prevalence of musicians' hand dystonia in the US orchestras would be 50-50% male-female, a clinical center limited to US orchestra players would see a ratio of musicians' hand dystonia patients of about 63 to 37% male-female, which incidentally is close to the clinically reported ratios. Male-female ratios of players differ significantly per instrument. For instance, flamenco guitarists are predominantly male. Consequently, the population of flamenco guitarists with hand dystonia reported by clinical centers will be predominantly male. Here presented are the statistics of the hand clinic for musicians at the Department of Plastic and Hand Surgery of the Erasmus University Rotterdam in the period 1999 to 2000. Following my appointment as a researcher in biomechanics of reconstructive hand surgery and musicians' hand dystonia, plastic and hand surgeon Jan Sonneveld started a hand clinic for musicians. Hand factors in hand dystonia cases were analyzed in my hand research lab. In these 9 years, the hand clinic saw about 350 musicians with hand complaints, of whom about 50 with hand dystonia, 14%. Of those, 79% were professionals, 8 and 13% were semi-professionals and amateurs respectively. Amateurs play in total much less than professional musicians, but typically practice or perform irregularly in bursts, for instance in weekends, have little time to maintain their playing technique, and also may tend to play repertoire somewhat too advanced for their techniques, which may also put them at risk of task-specific hand dystonia. This statistic shows the correlation between affected hand site and instrument type. In keyboard players, almost all dystonias are in the right hand. In string players, violin and cello, it is the left hand. In flute, the left to right hand ratio was 4:1. This ratio is also present in larger series reported in literature. In absolute numbers, the right hand guitarist dystonias equaled all keyboard players together. However, there are many more keyboard players than classical guitarists, so the prevalence of hand dystonia in guitarists is much higher than in keyboard players, likely well above 1%, and therefore not a rare condition. In the dystonia model published in Biological Cybernetics, biomechanical hypotheses were presented to explain the high left-to-right ratio in flutists and the high prevalence in guitarists. A brief review of musicians' hand dystonia research. Genetic associations have been identified in dystonias affecting general locomotive control. However, in task-specific musicians' hand dystonia, so far no consistent gene associations were found. A recent survey found in 50 of 367 musicians' hand dystonia cases, 14%, a family history of movement disorders such as dystonia, for instance writer's cramp and musician's dystonia, tremor, and Parkinson's syndromes. To facilitate a systematic review of research, a schematic of motion control in playing is presented. In the CNS, voluntary motion control is deconstructed into muscle activation signals. These signals are conducted from the spinal cord by the peripheral nerves to activate groups of muscle fibers in muscles, of which the combined activity produces muscle forces and length changes that are carried by tendons over joints, causing joint motions. The joint motions move body segments of given shapes, resulting in locomotive movements. Playing movements interact by contact forces with the hand instrument interface, activating instrument mechanics that produce sound. Forward control of motion stops at muscles, purple arrows. However, feedbacks go much deeper, blue arrows. Sound is fed back by the ears and movements by the eyes by dedicated nerve systems. The locomotor feedbacks pass through the peripheral nerves. Hand interface contact forces are captured by various touch sensors, and proprioception feedback is provided by various sensor systems in muscles, tendons, and joint ligaments. Control consists of modulation within the CNS of the signals leaving the CNS. Once the control signals leave the CNS, all processes they cause in the periphery are involuntary, consisting of electrochemical or mechanical transductions. In research of musicians' hand dystonia, peripheral nerve conduction problems as may result from nerve compressions were investigated. Some association was found between affected ulnar fingers and ulnar nerve pathology, for instance, ulnar nerve compressions at the elbow. At the Hand Clinic for Musicians in Rotterdam, a case was seen in which playing-specific dystonic symptoms developed in early stages of multiple sclerosis. However, such findings were not systematic. Muscles. To the degree that muscles work normally in other tasks, they are by themselves not likely pathologically causal in musicians' hand dystonia. However, muscle overuse, for instance fatigue and resulting physiological feedback, may be involved as will be discussed in the model. Central nervous system. The main research in musicians' hand dystonia has involved the central nervous system. However, so far, no CNS parameters have been identified that consistently discriminated task-specific musicians' hand dystonia from non-dystonic control musicians. Biomechanical hand factors. In parallel with and unrelated to our own biomechanical and anatomical hand research in musicians' hand dystonia in the 80s and early 90s, others have looked at correlations with musculoskeletal factors. Wagner and collaborators looked into the correlation between limitations on hand-finger joint ranges and overuse in musicians. They noted some correlation between limitations in MCP joint abductions and musicians' task-specific hand dystonia. Since Wagner and accepting our own research, musculoskeletal factors have not been systematically considered anymore in musicians' hand dystonia. Wagner's research was in line with the model here presented. However, the parameter set investigated by Wagner and collaborators was only a small subset of the possible peripheral risk factors considered in the present model. A brief review of current treatments. Neurologically, different approaches have been tried. Botox is being used to chemically block the innervation of overactive hand muscles. The aim is symptom reduction, which is sometimes helpful. However, Botox has significant problems. The effect is not permanent and injections must be renewed after a few months. There is the problem of determining what muscles to inject, of dose optimization, and of precise repeatability of injection points. Recently, awareness has arisen that long-term Botox use might damage injected muscles by causing irreversible atrophy. Retraining of movement and of proprioception has been attempted to improve the resolution of the sensory motor hand-finger cortex representation, which brain plasticity studies found blurred in musicians' hand dystonia. However, subsequent studies found the distinction with non-dystonic musicians less obvious. Therapeutically, the results of these training methods are not clear. In generalized dystonia, deep brain stimulation has achieved some spectacular results in suppressing control dysfunction. Extrapolating these results, deep brain stimulation electrodes have been implanted in a number of musicians with task-specific hand dystonia. These risky procedures will be discussed after the presentation of the peripheral factor model. There are several centers, among others in Spain, that offer motion retraining in musicians' hand dystonia. Many other paramedical centers or persons may see musicians' hand dystonia cases. Detailing this field is not within the scope of this lecture. Musicians with hand dystonia occasionally return to playing, testified by videos online, but the overall results remain vague if reported at all. A special group of musicians' dystonia treatment providers are musicians with dystonia who retrained themselves and became active on the internet in providing advice on how they managed this. However, the outcomes of such online advices are not systematically followed up and published. In conclusion, it can still be stated that no musician's hand dystonia treatment has proven systematically effective, that the outcomes are unpredictable, that is, a treatment may improve some cases but not others for reasons unknown, and that full recovery remains rare. Recovery is also not well defined. As a golden standard, I propose that full recovery means the resumption of playing activities at least at the performance levels prior to symptoms without fear of relapse. As general conclusion of research and treatment, the following. No individualized CNS causes or predispositions have so far been identified in musicians' task-specific hand dystonia. Equally, no systematic peripheral factors have been identified, although incidental factors may be present. Extrapolating, in musicians' hand dystonia cases that recover, systemic CNS causes are unlikely. Conform with the peripheral factor model here presented, some neurological etiological models now propose non-pathological learning processes in conducive environmental circumstances. After this brief review of research and treatment, an etiological multifactorial model will be presented of how peripheral factors might be crucial in developing task-specific musicians' hand dystonia. The model is based on the following observations. In the deconstruction in the CNS of a voluntary motion control into muscle activation signals, defective processes can occur at many levels. However, defective processes in the CNS should produce generalized symptoms. The present model assumes that the more task-specific the symptoms, the less likely that CNS defects are causal. For example, some pianists can play symptomless with the affected hand by using only 4 fingers. Symptoms only occur when they include the remaining finger in playing. It is hard to imagine a defective systemic CNS process that would produce symptomless 4-finger play and symptomatic 5-finger play. Therefore, the peripheral factor model assumes that in such cases, the crucial causation factors do not exist in the CNS, but in the peripheral task environment, and that the development of the control dysfunction is driven by task-specific feedbacks from this peripheral environment into the CNS as a controller. This leads to the following hypothesis. 1. CNS function in task-specific musicians' hand dystonia can be normal. The more task-specific, the more likely that there are no CNS predispositions. Therefore, by hypothesis, the CNS in most task-specific musicians' hand dystonia is normal. 2. With normal CNS, task-specific musicians' hand dystonia results from normal learning processes, not pathological processes. 3. If both these hypotheses hold, individual involuntary factors are generally necessary cofactors for developing task-specific musicians' hand dystonia. 4. Intense playing repetitive behavior is also a necessary cofactor, but if hypothesis 1 and 2 hold, by itself insufficient to cause task-specific musicians' hand dystonia. 5. CNS factors are by no means excluded, and any intrinsic CNS control problem will contribute. However, we hypothesize that in the majority of task-specific, musicians' hand dystonia cases, CNS predispositions are not the cause. Some observations of task-specific musicians' hand dystonia versus writer's cramp. In past studies on focal hand dystonia, musicians' dystonia and writer's cramp were often lumped together. Therefore, it should be stressed that musicians' dystonia and writer's cramp derive from very different populations. Writer's cramp recruits unfiltered from the world population as everybody learns to write. Therefore, CNS dysfunctions such as tremor might not be rare in writer's cramp. For instance, with tremor, one can readily imagine that larger than necessary forces are used to grasp the pen and to increase stiffness in the muscle tendon system to enhance pen control accuracy against the tremor. In contrast, professional musicians are a highly selected population in which CNS dysfunctions such as tremor would be incompatible with acquiring professional performance skills. In fact, musicians likely constitute the most severely selected population for finger control accuracy existing. Therefore, neurological findings in writer's cramp cannot be extrapolated to musicians' dystonia. Musicians' dystonia and writer's cramp populations should be considered as fundamentally different and should not be mixed in studies. This implies that the peripheral factor model here presented should not be critiqued by results of writer's cramp studies. The key questions of the peripheral factor musicians' hand dystonia model. Can control problems result from peripheral factors and by what processes? In answer, a factor analysis of instrumental playing is presented. Acquiring a playing technique is a movement optimization problem in which 3 factor sets must be harmonized. 2 sets are involuntary. One consists of the individual neuromusculoskeletal properties of the player, which may differ between the hands. Factors are hand dimensions, hand-finger joint ranges of motion, individual muscle tendon anatomy, properties such as joint laxity, congenital or traumatic defects, and others. These factors constitute or limit the set of feasible motions. The second factor set is constituted by the hand-instrument interface, its layout, the key resistance forces, depth of key travel, and others. This set determines the minimum of movements required to play the instrument. The third factor set contains all voluntary factors: the playing technique, performance level, repertoire choice, practice intensity and methods, performance program, and others. These voluntary factors constitute the occupational load of the locomotor system. As an example of hand factors, let us look at the individual variability in the anatomical independence of finger muscles and tendons. Anatomically, muscles consist of muscle bodies and tendons. Muscle bodies consist of muscle cells that contract when nerve signals arrive. Tendons are strong, rope-like structures connecting muscles to bone and transferring muscle actions to joints. Each finger has 2 flexors and 1 or 2 extensors. The extensors are the muscles that lift the fingers. Their anatomy is as follows. The extensor muscle bodies are slender and lie at the backside of the forearm. Their tendons cross the wrist and run over the back of the hand to the fingers. All fingers except the thumb receive tendons from a muscle group called a common finger extensor, anatomically called extensor digitorum communis. The index and little fingers each have an additional extensor. The index extensor lies at the deeper anatomical plane not visible in this picture. The little finger extensor has a slender muscle body in the forearm next to the extensor communis. For the ring and middle finger, the extensor communis is the only extensor. The tendons of the common finger extensor are anatomically not independent. Near the fingers, these tendons are interconnected by tendinous connections called the juncturae intertendinei. These interconnections play a role in stabilizing the extensor tendons over the base joints of the fingers, called the metacarpophalangeal joints. The junctura are congenital, it is present at birth. While having general characteristics, these interconnections are quite variable between hands and persons, forming in each hand a unique pattern. The junctura limit the independence of the tendons they interconnect. Being of tendinous material, they cannot be permanently modified or lengthened by exercise. The tendon of the extensor indicis, which lies at the deeper anatomic plane, is not interconnected. The extensor digiti minimi is interconnected to the juncturae system by the insertion of the tendon of the extensor communis to the little finger. Here is an example of how juncturae can limit or even exclude independent finger extension. In hand A, the ring and middle finger extensor tendons are interconnected, but the tendons themselves are independent. In hand B, the little ring and middle finger extensor communis tendons form a single tendon plate. As a result, independent extension of the middle or ring finger is not possible. Moreover, no amount of practice can modify this anatomy. Such a hand lacks by birth the finger independence that is necessary to acquire professional pianistic playing skills. In conclusion, not all hands are, for anatomical reasons, capable of developing professional musical playing skills. In the hand lab, I designed with collaborators tendon actuators to simulate realistic motion in anatomic hand and finger specimens. The finger tendons are connected by highly inextensible ropes to motors or manual actuators that produce forces and tendon motions like real muscles. The tendon forces are measured by load cells and the tendon motions by displacement meters. The tendon actuator device allows demonstrating how the extensor tendons move in reality. This video shows extension flexion of the middle finger while the index is flexed. The fingers are splinted so that only the MCP joints can move. In full extension, the middle finger extensor tendon forcefully pulls the index extensor tendon by the interconnecting junctura. In contrast, the junctura between the middle and ring finger tendons remains slack and therefore does not interfere with any tendon motions. Overall, pianistic playing motions are smaller than the motions in the video. Therefore, in reality, in this specimen, the junctura between index and middle finger extensor tendons would not limit pianistic playing motions. In this video, the ring finger is lifted relative to the little and middle fingers. The junctura to the middle finger and to the little finger then become immediately stretched and hold further ring finger extension. Such limited ring finger extension may be problematic in piano playing. In conclusion, juncturae form limitations in playing only when they limit the independent tendon motion ranges required to play the specific instrument or instrument type. Therefore, the same junctura may be more or less limiting to playing motions depending on the differences in layout and finger movement ranges of the hand instrument interfaces. In the classic anatomy, the common finger extensor is considered as having limited independence in the muscle bellies to the individual fingers. However, the detailed studies here cited demonstrated both by anatomical dissections and by functional surface EMG measurements that the extensor communis muscle bellies to index, middle and ring fingers are generally anatomically quite independent, even if their tendons are interconnected by junctura. The pictures show the general internal anatomical structure of the extensor digitorum communis and the anatomically optimal EMG surface electrode positions for measuring independent EMG activity in these muscle bellies. In the deep finger flexors, anatomically called flexor digitorum profundus, and the long thumb flexor, called flexor pollicis longus, interconnections between the finger tendons are also not infrequent. These muscles flex the end joints of the fingers and thumb. Their powerful muscle bellies form a deep layer occupying the entire forearm. Their tendons cross the wrist through the carpal tunnel and insert in the fingers and phalanges. This picture shows a dissection of the deep finger and thumb flexors. The tendons are pulled out of the carpal tunnel at the wrist and spread apart to show their interconnectivity. There is a strong tendinous connection between the thumb tendon and the index deep flexor tendon. There are tendon fibers from both the middle finger and from the little finger tendons inserting into the ring finger tendon. Therefore, flexing the thumb end joint will flex the index, and flexing the middle finger or little finger end joints will flex the ring finger. These tendinous anatomical interconnections cannot be modified by exercise. This picture shows deep finger flexor muscle bellies to the different fingers. In some anatomic specimens, as in A, the deep flexor muscle parts to the different fingers have gliding layers in between that allow them to be easily and cleanly bluntly separated. In other specimens, as in B, such a clean anatomical separation of the deep flexor muscle parts to the different fingers is not possible. In B, the muscle parts to middle, ring and little fingers form a mass that cannot be well separated, and also tendon strands cross from one tendon to another. In these cases, it is doubtful if, even by training, a muscle part to one finger can be activated without activating some muscle fibers inserting into the tendons of adjacent fingers. In contrast, it is likely that anatomically well separable deep flexor muscle bellies, as in specimen A, could be trained by musical practice to be activated fully independently. The next slides present some more examples of tendinous variations limiting independence in the deep finger flexors. Such variations are not exceptionally rare. Here shown is a middle finger tendon that received two tendons from separate muscle bellies. However, one of these tendons split and about half of its fibers inserted into the ring finger tendon. Similarly, about 1/3 of the deep flexor tendon of the little finger also inserted into the ring finger tendon. This specimen shows a ring finger deep flexor tendon that divides into two, with about half of its fibers inserting into the middle finger deep flexor tendon and the remainder half forming the ring finger deep flexor tendon. The little finger deep flexor tendon inserts about 1/4 of its fibers into the ring finger tendon. LU1 to LU4 are the lumbrical muscles. In summary, deep flexor tendons can be independent as in A, are completely fused as in C, with all variations in between. Remarkably, variations as in C do generally not cause problems in daily tasks. Usually, the superficial flexors are more independent, which allows to compensate for deep flexor interdependencies, and daily tasks generally do not require great finger independence. However, If an instrument type requires the independent use of the deep flexors, case C would not be able to learn to play this instrument at a professional level. Some further information on variability in deep flexor independence and more general information on the structure of muscles in the forearm can be found in the papers here referenced. In summary, the list of hand factors that may negatively impact instrumental playing is long. There is a large inter-individual variability in anatomical independence of finger muscles and tendons, ranging from full independence to fusions. Other anatomical limitations or problems may exist, such as limited joint ranges, congenital or acquired by trauma or disease, post-traumatic joint stiffness, and others. Tendon gliding may be diminished, for instance by scar tissue. Joint ligaments may be lax or subtly damaged without significant effects in daily tasks. Peripheral nerve signal conduction may be compromised by nerve compressions or systemic diseases. Morphological features may cause ergonomic problems at the hand instrument interface, such as hand size, relative finger lengths, or small joint deformities causing unergonomic malalignments. And then there are many other factors possible. In terms of effects, peripheral factors produce distinct categories of problems. Too many limitations on motion or finger independence may produce hands that are over-constrained relative to the playing task so that no optimal playing motions are possible. Instabilities in joints or muscle control defects may create under-constrained systems in which playing problems arise from mechanical controllability issues. Energy losses due to increased soft tissue stiffnesses or increased tendon gliding friction may cause various forms of muscle fatigue in repetitive motions. Dimensional hand interface incompatibilities may increase playing efforts and with it the likelihood of various forms of overuse. Nerve compression and peripheral nerve pathologies may weaken muscles and decrease activation accuracy. To conclude the review of the parameter set determinant of feasible motions in the model. Peripheral factors may, among others, exclude functional playing motions, increase playing effort, cause control instabilities in the joints or control problems in muscles, and cause geometric incompatibilities. Moreover, the effects of simultaneous factors are cumulative. Lecture 1, end of part 1.